Takes those seriously. And students take those seriously, by the way. So let's go ahead and quickly go through the perimeters of the purine metabolism. All right. Of course, we start from bicarbonate, and this is for pyrimidines in this case. We start from there, and we go all the way to aspartate. All right, and then aspartate will take us afterwards with carbohydrate phosphate all the way to our first pyrimidine, our first nitrogenous base, orotate. Now, the question, one question that can be asked about the first two reactions in the pyrimidine biosynthetic pathway. All right, what do those look like? Okay, what do those look like? The first two reactions of the pyrimidine pathway. Okay. Now, of course, well, let's answer it. All right. All right. Okay. So it is the urea cycle. It is not the TCA cycle. So this is very important. Now I can easily ask the same question about the purine biosynthesis. Would it look like the urea cycle, the first reaction of the purine biosynthesis? Would it? No. It's only pyrimidine. Yeah, the first two reactions. As a matter of fact, we have an isozyme, which is the so those are the two reactions, and so this is the first one. And who is the nitrogen donor for the first reaction? Is it free or is it glutamine? That's right, it's glutamine. Okay, so glutamine donates the nitrogen. Of course, we need 2 ATP here. And then aspartate transcarbamylase, which is olefin transcarbamylase, similar to the same kind of reaction where we're building our big molecule. From carbamyl aspartate, we go all the way to rotate through two reactions, okay? Hydration and reduction. And that gives us our first nitrogenous base, the first nitrogenous base, which is orotate. Now orotate, from there we can go to one more bottleneck, which is uracil, our first official nitrogenous base, our first pyrimidine, to be more accurate. And then from pyrimidine we can build all the other pyrimidines. So the first one is UMP, uracil monophosphate. And of course, we've seen the difference. And remember, I asked you to always keep those structure in mind in order to get to those reactions. You know, methylation, carboxylation, amination, etc. So anyway, this one is carboxylation, and that gives us, of course, or decarboxylation rather, that gives us our uracil. And then from uracil, of course, we can make cytosine, and we can make. All right. And we also looked at the reactions that give us the di and the triphosphates, and those are kinases. Kinases are enzymes that add phosphate. Okay? So really, pretty much we can exchange any di with the triphosphate, and the phosphate can simply jump to the other. CTP synthase, that's the cytosine triphosphate, cytosine triphosphate, or CTP. That, of course, needs amination. Glutamine, again, is our donator, and ATP is used for this. And then for the enzyme ribonucleotide reductase, and this is really the gateway between the RNA and the DNA, okay? Or the oxy versus the deoxynucleotides. This enzyme works on all of the regular nucleotides to give us the deoxy, and the donor, of course, of the high energy electron is NADPH. Now, of course, with NADPH and with ribose, we've seen the intimate link between glucose phosphate pathway and nucleotide biosynthesis. Now, for the purines, of course, we went through the anatomy or the source of every atom of the purines, and we've seen that obviously it's a bigger ring, so it has more sources. So carboxylation, glycine, aspartic acid, and glutamine are the three amino acids that contribute to that. Activated carbon is carried, two of them actually are carried by, again, tetrahydrofolate, and of course, one carboxylation. Now, we mentioned that we need to be aware of the very first reaction, because Purines are built one atom at a time on the ribose, right? They are assembled right there, they're built right there. And so the first one, the first precursor, is the ribosine amine, all right? And again, the donor of the amine here is glutamine. One more time, glutamine, glutamate amine, and that gives us the 5 ribosine 1-amine. Now from there, we have several reactions we need to worry about, but our last product, our first purine will be inosinate. Okay? Now from inosinate, we can build the adenine or the adenylate as nucleotide as guanylate, and we mentioned those 2 reactions, this 4 for them, from inosinate to adenylate and to guanylate. Now, the many things that are important. One of them, of course, is our direct link with the citric acid cycle through fumarate, and that goes with the adenylate. And that enzyme, of course, adenylate or adenylosuccinate synthase. And we mentioned something very important here as a reciprocal control of both pathways, which I'm going to mention again in a little bit. Basically, the source of energy, both reactions need energy, right? High energy phosphate equivalents, ATP or GTP. So GTP happened to be that source of energy for making ATP, and ATP happened to be the source of energy that's required to make GTP. Okay? So if one of them is missing, the other one will definitely not be made. Okay? So this is one of those cases where we have strict specificity here substrate. This enzyme will not work with GTP. With ATP. Will not accept ATP as a source of energy. Okay. This is the ribonucleotide reductase, the one I just talked about. Now that is a very important controlled gateway for this pathway, for that direct DNA pathway. And its final product, one of its final products, which is the deoxy-ATP, is an inhibitor, while ATP, which is the oxy version of this. Where is it? Oops. This time, right? Those controls are extremely important. So you can see there are so many levels of control. Of course, we have purines and purines in the first place. So we have 2 major pathways. And then we have common control to both of them and specific control to either one. Okay? In blood, and they are formed inside the mitochondria. They are formed inside the mitochondria. As a matter of fact, they are formed inside the mitochondria in the liver specifically. Okay? Those are the basically 4 reactions that make our ketone bodies. All right? And notice they start from acetyl coenzyme A. Okay? So we have the first reaction, which is a parallase, which is the same enzyme that is part of the beta oxidation, that gives us the acetyl coenzyme A. And that's just a fatty acid, very short fatty acid, 4-carbon fatty acid. And from there, of course, it's 2 ketones, so it's really the last part of the beta oxidation of the 4-carbon fatty acid. And then from there, we make the intermediate, the glutarate coenzyme A, the hydroxymethyl glutarate coenzyme A, that eventually becomes our acetoacetate or our first ketone body. Now, our first ketone body, we can make the other 2 ketone bodies from there, which is the hydroxyglutarate through this enzyme and the acetone. The acetone itself does not need a reaction, does not need an enzyme. It's a reaction, but it doesn't need an enzyme, and hence it happens. And so we have ketones, we have the synthase for the glutarate, and then we have hydroxymethyl glutarate coenzyme A, and that's the one that gives us the acetoacetate. So it's a cleavage reaction because we add 3 acetyl groups and then we end up getting rid of 1. And the idea is just to produce this unfavorable reaction. Like the energy we use is we use it from the coenzyme A there. So because of that, you think this reaction is reversible or not reversible? That's a given question. Do you think it's reversible? We put that much energy in it, which is very similar to ATP. Would it be reversible or not reversible? I'm sure the arrow is telling you something as well. All right, we still have people that believe it's reversible. This reaction is very irreversible. Yeah? It is irreversible. As a matter of fact, the reason it's irreversible is because we don't want this to come back when we actually leave ketone bodies. Okay? We have another enzyme that will take us back. So this reaction is reversible, and that is the reason that we will come up when we talk about the going back to use it as energy. Now, the hydrogenase, the hydroxybutyrate, the hydrogenase is the one that gives us the hydroxybutyrate. Okay? Now, as far as energy is concerned, everybody has to go back. All right. So to use ketone bodies for energy, they have to ultimately, and this is the question here, ultimately convert to what? We cannot choose acetone, we cannot use butyrate, and we cannot choose acetoacetate straight away. You're going to have to convert it to which one of those things? You have to convert it to one of those. All right? And then it can go through a regular energy production after that. But the key one that we have to convert to, which is the reason why the reaction is irreversible, is, what is B? Acetoacetate, absolutely. Absolutely. Acetoacetate. It's not acetoacetate, we cannot use acetoacetate. Of course, it's part of the path, but ultimately we have to get to that intermediate that is similar to the beta oxidation. And that's acetoacetate. Glucose, of course, as we mentioned, at least as far as this goes is concerned, we cannot make glucose from that. All right. So, okay. Which enzyme, and here, that's not a tricky question, by the way, it's a real question. Which enzyme produces acetoacetate enzyme A when we are what? When forming the ketone bodies. So we have acetoacetate enzyme A produced by two ways. But when we need to form ketone bodies, which one of those enzymes we use? When we are forming the antibodies, we are making ketone bodies. All right, ready A. That's coenzyme A transferase. That's coenzyme A. When we're using it for energy during starvation, is when we use that enzyme. So there are two paths here, and here's what I need you to know. This is acetoacetate coenzyme A. We can produce it this way, as you can see this arrow here. That's from 2-acetyl coenzyme A, right? Who's the enzyme for this? Thiolase. All right? That's when we're forming ketone bodies. When we are using ketone bodies for energy, we cannot use this. We have to use the coenzyme A transferase. Okay? So the question, the exception here might be when we are forming antibodies, or when we are ketone bodies, or when we are using them as energy. Okay, coenzyme A transferase utilizes, it needs the coenzyme A to actually put it in that acetoacetate, and that comes from succinate coenzyme A. Okay, succinate coenzyme A is the donator for the coenzyme A, and that gives us acetoacetate coenzyme A. Now here, this is a beta oxidation intermediate, so the rest is basically cleavage, and after you break it with thiolase, then we can go to the citric acid cycle to produce. The question is, ketone bodies are produced only during starvation, or do we need them in other stuff? Yeah? Are ketone bodies produced just for starvation? Is that the only time we produce them? We don't need them at all, unless we are starving? Or do we use them in our everyday? All right, so it's false, absolutely. Ketone bodies, I might have given that wrong impression yesterday that ketone bodies, when we finally have no more energy, especially for the brain, we use ketone bodies, and that's true, but we actually make ketone bodies every day. Okay? We just make much more of them when we are in starvation. As a matter of fact, Matter of fact, the heart and the kidney, they prefer ketone bodies as a source of energy over glucose. The heart and the kidney muscle, they do prefer that over glucose. The brain, however, will take glucose and will not take anything else until there is starvation. And so that's where the concept of starvation came from. But otherwise, we actually do produce ketone bodies. A ketogenic diet, the idea of it is to produce more fat, right? To where we force actually more production of ketone bodies in the blood, hence using the fatty acid as our source of energy. Okay? Which of course has its own problems and caveats. But actually the heart and the renal tissues will use that. And so this is very important. So ketone bodies are not bad. They're not associated with bad things all the time. Yes, sir?
So when we do talk about ketone body, we do starvation. Do our body use amino acid then?
You what?
Do our body use amino acid when the ketone body is like completely depleted?
They're usually not, they're the last thing to deplete during starvation.
So how long do we have ketone body if we don't consume any more food?
How long?
Yeah, can do we like
After starvation?
Yeah, after starvation.
That depends from human to human.
Because of the body fat?
Because of the body fat. Now, I bet you I can survive longer than you. That's it. I mean, as funny as it might sound, but it depends on the person, right? So it's because and then of course how much the body can tolerate it too, because they're not designed to be flooding the circulatory system. Okay? So when you want to, so yes, it depends. So some people can last really long time on ketone bodies, and some cannot. Okay. All right. Okay. So here we have the liver, and this is during fasting or diabetes, which is something we're going to be talking about next week. And so what happens here, the fatty acids now are broken down from the triglycerols, that we know that already, and the glycerol will immediately be used in gluconeogenesis to give us glucose. All right, but we have only one molecule per 3 molecules of fatty acids, and hence we have far more acetyl group than glycerol, obviously. And so those acetyl groups will then end up going into the ketogenesis, making the ketone bodies from those enzymes we mentioned, and then that can, of course, go through the blood. Of course, the heart is already using it, prefers it, that's fine, but the rest will end up going to the brain. Okay? So here, of course, the fat cells are the one that give us those, but the brain is not shown here. That's the one that can use that. Of course, we can use it as long as we have acidity going. Of course, again, and again, the reason is glyoxylate cycle is something we don't have. It's the cycle that makes glucose from acetyl coenzymes. We cannot do that. Well, as I mentioned yesterday, of course, research has shown otherwise, but at least for the purpose of this course, this is what happens. Now, we're going to go into signal transduction. Why signal transduction? And this is actually going back to chapter 14. And we're going to talk about it superficially, however, there will be some specific information that's very important. Why signal transduction, and why do we really care? Well, a bacteria, or single-cell organisms in general, when they are faced with any environmental changes and environmental calamity, it's one cell. It will sense whatever is happening and it will change. Multicellular organisms, such as ourselves, we have several tissues. Now we have really these specialized tissues. All right. So when an environmental thing happens, be that starvation, extra food, extra alcohol, etc., cold, fear, then we have to have a way to communicate, because all the tissues have to work in tandem, right? We cannot afford things working, like out of control, not based on that hierarchical system. Okay? So we have these different sensors, including, by the way, our eyes, ears, and skin, that senses stuff. And of course, inside we have molecular sensors that can detect all kinds of chemicals. Very, very important sensors, very sophisticated sensors. So based on that, a message has to actually be initiated from whatever tissue that senses something, and it has to be going through all the tissues, the other tissues, in order to give the message. Okay? So we need to have a primary messenger. Usually those are hormones that can be either peptides or just simple chemicals, and those can travel and then they have to be received by a tissue. All right? Reception by the tissue is very important, but then this message has to be actuated. Okay? So, any environment that happens, usually we have 5 major steps for the communication between all tissues in the body. As a matter of fact, it's a horrible disease to have if you don't have this communication going on in different tissues. So the first thing is the primary messenger, which is basically by whatever sensor that detected something that happens, like high blood glucose, for example. So insulin, for example, would be dispatched. That's our primary messenger. Now that has to be received, and has to be received well. In other words, when it goes to the cell, it cannot necessarily go inside the cell, but it has to make the cell know that I'm here as a messenger. So it has to have protein or receptor on that membrane of that cell that will allow it to come. And those receptors, by the way, are specific, okay? So they can bind only through them, to these primary messengers. And again, the binding usually is through weak bonds, like hydrogen bonds, ionic interactions, but they're specific, nevertheless. Now, once that's received, the enzyme in the cell has to know that there's a message that is outside the cell. And so we're going to have something called a relay information that a secondary messenger has to be produced. The secondary messenger usually always inside the cell, And that secondary messenger then, the job of that secondary messenger is to amplify this message. Yeah? We're talking about one molecule that's going to the cell that has to be amplified to thousands of molecules and many biological effects, including many of the reactions we talked about throughout the semester. Okay? So that's the amplification. And that's when the actual message is actuated, the physiological response comes to play. The metabolic pathway is activated. The metabolic pathway is inactivated. That's the kind of thing that happens. And then, of course, we have to terminate that signal. Because not terminating the signal is really as bad as not having the signal at all. So, the mechanism to terminate the signal. We're going to be talking briefly about three major types of these communications, signal communication. Okay? And you will see that there's no pathways, because we're going to talk about chemicals, but we are definitely going to be talking about many enzymes and many events. You'll see one of the underlying themes is called variant modification, which is phosphorylation, biphosphorylation specifically. So the phosphorylation that we've been talking about since glycolysis as an immunocontrol, we will see actually far more of that happen during the signal transduction, phosphorylation. And we will see a lot of conformational changes, a lot of 3D changes, and hence a lot of weak bonds breaking and reforming in order to get this message to go through. Which is, again, a testament of how the sequence of the protein is been structured by the right flow and the right conformation changes. So, here are some examples of secondary messengers that are inside the cell. Cyclic AMP and cyclic GMP in front of it. And so this is basically the nitrogenous base here, right? And we have the ribose that actually form a cyclical relation with the phosphate on the carbon number 5. Calcium is very important secondary messenger. We see as many, we haven't talked about it much as a matter of fact at all, but for example, in muscle contraction, calcium has a very important role to play in order to allow these things to happen. So the actuation of that message happen there. Inositol, phosphate. We've seen inositol briefly when we talked about phospholipids, right? Now, I didn't ask you to memorize the structure of inositol then, and I'm going to ask you again here. I need to be familiar with or aware of inositol. Okay? Diacylglycerol. Now, we are familiar with diacylglycerol. Are we familiar with triacylglycerol, triglyceride? You're familiar with that, right? So diacylglycerol is just 2 fatty acids to a glycerol. That's part of the membrane. So even part of the membrane can serve as our secondary messenger. All right? Secondary messenger can be anything that will ensure quick communication inside the cell. Okay? So while we start with one molecule, such as a hormone, we will have thousands of molecules as a secondary messenger. That will reach tens of thousands of different enzymes that will do millions of reactions, or millions of molecules in each role. Okay? That's amplification, that's a cascade. Okay, the first class of single transduction pathway, is the 7 transmembrane helix receptors. It's a family of proteins, receptors, that means they are on the membrane of the cell, and they receive the primary message. Okay? So those are 7 transmembrane helix receptors because they have 7 alpha. And those 7 alpha helices, if you count those 7 alpha helices, those are the ones that span the bilayer, the membrane bilayer. So that means they are made of what? The R groups are hydrophobic amino acids that are interacting. So they have that transmembrane domain, which is something we expect, right? Because it's on the membrane. So since it's a receptor for a hormone, that means it has to have an outside domain that will receive, that will recognize that, and that will be somewhere here. Okay? So these random folds that you see here, those loops, are actually perfect fit for whatever hormone that's going to come. No other hormones can bind here. On the other hand, down deep after the transmembrane, the cytosol, we have also, of course, the domain as well. Because it has to receive that, the message has to propagate through that, right? Otherwise, what's the purpose of having a receptor that will not give the message to the cytosol? So there will be actually conformational changes here too. So strict conformational changes by binding of one molecule is the one that can start actuating the message. It's credible. Weak bonds. Okay? All right. Some of the receptors are the beta adrenergic receptors. Actually, those are the ones for adrenaline. That increases heart rate and other metabolic events, actually. All right, so those are the penetrant, adrenaline, one of them. Conformational changes, again, is the most important part of this. The right sequence, the right fold, and the right changes. All right, another player in the 7TM receptor, the 7 transmembrane receptors, is the G protein. Yeah? This is what the G protein looks like, and you can see the elaborate 3D structure here between the beta and the alpha. I need you to focus on the cartoon for the simplified look. It has 3 domains, alpha, beta, and gamma. If you look at the alpha actually is a hydrolase, a GTP hydrolase. It can bind GTP, and it can bind GTP. If it binds GTP, we're going to see triphosphate. It will hydrolyze it, we're going to see diphosphate. The binding of GTP or GTP is strictly based on its 3 conformational structure. Again, the fold plays a role. It cannot bind both of them. When it's in a certain shape, it will bind one of them. When it's in another shape, it will bind the other. Very important. These are all important background. All right. The other player is adenylate cyclase. That's the enzyme that takes ATP and makes a cyclic ATP. Cyclic ATP. Then AMP, right? That's the one that produces the secondary messenger, the one that goes into the cytosol and activates other things. We're not very interested in the other things that's activated. However, we do know some of those other things that activate. Because it's a very big cascade. Okay? It's a semester long, more than a semester long course of study. All right, adenylate cyclase. This is of course how it looks like. That's fine. And this is the overview of what happened. Yeah? So, the epinephrine, for example, will activate the receptor by conformational changes. The activation of the receptor is going to lead to the activation of the G protein that eventually will activate the adenylate cyclase that eventually will produce cyclic AMP. So you see, it's like a tag game. You tag, you're it, you get active, you go and do whatever you have to activate, and so on and so forth. With every activation process step, more molecules get activated, and so you get the cascade. All right? You start from 1, you end up 2, 4. Of course, it's far more liberating than that. All right. So this is, of course, some of the stuff that epinephrine do, right? Part of the result of this communication, where you have more acid in the stomach, you have more chloride channels open, pigmentation change, respiration, etc. But again, the main part. So let's look at this in a cartoonish way. So here's our 7 transmembrane, 7TM, and the hormone will bind here. All right? And here, by the way, on the membrane, we have the adenylate cyclase. Adenylate cyclase, again, produces what? Cyclic AMP, our secondary messenger. And here, on the cytosolic side of the 7PM, binds our G protein, and it's alpha, beta, gamma. And usually, it's always, by default, bound to the GDP, to the diphosphate, by default. Now, when a hormone binds, what can happen is, it's going to change conformation, as you see, and it's going to bind GTP. It will spit out GDP and bind only GTP. But when that happens, it will actually itself undergo conformational changes. When that happens, those conformations, it can no longer bind to that hormone, to that receptor. And so it will leave immediately and disperse, leaving behind it the beta and gamma. It will disperse in that cytoplasm until it finds adenine cyclase, and then it will attach very strongly by means of weak bonds. So it only can bind to adenine cyclase when it's in the GTP conformation. Once that happened, now adenine cyclase is active because it's bound to the GTP. When it becomes active, it will immediately convert ATP to cyclic AMP. So this enzyme is going to produce millions of cyclic AMPs that will go and bind to other proteins, including what? Protein kinase A. Protein kinase A will do what? Does anybody remember? Pyruvate kinase, eventually pyruvate kinase, phosphoryl kinase too, right? All those things. All right. So this is what happened, and many other things, by the way. Okay, so you can see where this started from. So when we go for glycolysis or the cancer cells try to do that, they try to actually work with this kind of level. So this is the ultimate authority for any pathway. They try to activate it this way. Does that make sense? There are several steps going on. They're simple steps, but there are several steps nevertheless. Okay? They all involve this kind of binding, conformational changing, activating the next one, activating the next one, etc. Okay? Now, how long is this message going to go on? Adenine cyclase is an enzyme, and it will just keep converting ATP to cyclic AMP. There's really nothing to stop it. As a matter of fact, the only reason it's going is because this is bound to it. So this is like the key that makes it active. So to stop it, really we have to kill it. Right? That's the only way we can do that, to activate it. Otherwise, it will keep producing cyclic AMP. Or we have to run out of ATP. So what happens is that GTP will be hydrolyzed to GDP, and once it's hydrolyzed to GDP, this is now the overall view, it will go back and bind up. It can no longer bind to the adenine cyclase. Okay? So we want to terminate this message, and that's the only way to do it. So really, as I mentioned here, the G protein actually, that one that binds to GDP, has an internal clock, if you will. It's a timer. It binds to GTP for a limited time. In other words, it hydrolyzes GTP to GDP very, very slowly. Okay? Just enough for the time to produce enough of that cyclic AMP. Once that time passes, then of course that GTP is going to be converted to GDP, and then of course it can no longer bind. It's not in a conformational shape to bind. It will leave and it will go back to its beta and gamma, binding to that hormone and leaving that energy. So it's a timer. It's not really a timer. And it's a good thing it's a timer too, because otherwise it will be active forever. As a matter of fact, some pathogens, bacterial pathogens, take advantage of that as well to do bad stuff. Is that all? Is that the only way to terminate it? No, we need more insurance. The message has to be terminated because, as you can see, many events happen, including producing enzymes. That's very bad if we don't terminate it, or make sure that it's terminated. The other one is actually on the level of the hormone slash 70M protein itself. Okay? We need to make sure that hormone doesn't bind to it again, at least for the foreseeable future. All right. Now, in chemistry, everything is in equilibrium, so that hormone is not blocking there forever. It's basically in some sort of equilibrium, and eventually it will dissociate. It will leave. Okay. But in that fraction of a nanosecond that the hormone leaves, a protein called beta-arrestin. It's arresting because it arrests. It will bind immediately to the cytosolic side, right here at the 7PM, and that will lock the gate and the outside part. And the hormone will, even if it exists, it will not be able to bind. So now we also killed the messenger. No pun intended. The messenger is still right, or at least the way the messenger is binding has been destroyed. And that happened also because of many conformational changes. All right, 70M. Are we cool with 70M conceptually? See how many events happening before all the level of factor we're talking about? What happens? Yes?
Okay, so GTP activates the GTP activation?
By activation and deactivation, can it bind to one and not bind to the other? Yes. So the hydrolysis of the GTP to GDP will make it not bind to adenine cyclase, but rather bind to the beta and gamma subunits.
Okay, so when it binds to the beta and gamma subunits,
that means it's no longer activating anything else. And that's the idea. All right, that's our first class. Very intuitive, hopefully, very similar. All right, now let's see who take advantage of this. And many bacteria take advantage of this. We have two kinds of toxins. Cholera toxin, we're very familiar with this. Actually, cholera toxin has an affinity to the G protein. Has an activity of the G protein, and it's a very strange affinity because once that bacteria produces that toxin that can go inside the cytosol, it will come here and it will lock its GTP conformation, preventing it from ever hydrolyzing GTP to GDP. What does that mean? That means this is going to be bound to adenine cyclase forever. Okay? And that means it will continuously make cyclic AMP whether there is somebody here or not. Okay? So it short-circuited the whole network. And hence, we're going to have a lot of protein kinase A, binding and activating a lot of stuff. And what's one of the results is all the ion channels will open, and we'll have very fantastic diarrhea until this thing stops. Okay? That'll be one of the things, because all the channels are open, and then you can lose water and stuff. Okay? Of course, like everything else, there is equilibrium, and of course, it will have to take time once you hydrate yourself properly. But that's one way the bacteria utilizes. Of course, now, consciously, I'm sure they're not aware of this network, but somehow it works the system to where it keeps bacteria. Pertussis toxin, the whooping cough bacteria, actually, its toxin binds to the GDP. Okay? And it locks it in that form. So no matter who comes knocking the door, it will never go beyond that. Okay? And that will eventually manifest itself in the whooping cough. Okay? So this toxin actually prevents the entire messenger the message to be relayed through a cell. So you can see that any of those biological events that's happening in our bodies and other nanozoology cellular organisms can be exploited somehow by something. If there is a biological event happening, there is a toxin that can exploit it. Or there is some sort of chemical that can exploit it. As a matter of fact, we use a lot of drugs this way as well in order to cure certain diseases. All right. So this is all that I want to talk about as far as the 70M. Now we have dimeric receptors. Dimeric receptors, those are not 70M. Those have two subunits. Okay, they're also obviously membrane. Receptors have to be membrane proteins, membrane part. And so they have, first of all, two parts to them, and they can never. Somebody fall asleep? All right, let's make it fast. So, oh my goodness. They didn't show. I don't have it. So that hormone, that receptor actually is also it's also of course binding to the hormone, but the mechanism of binding and activation is different. It's very different. It's fascinatingly different, as a matter of fact. We have an enzyme associated with this called a tyrosine kinase. Now, right off the bat, if I ask you, a tyrosine kinase, what do you think this enzyme does? It phosphorylates tyrosine, right? Serine kinase phosphorylates serine. Okay? Those are part of the modification enzymes. Okay? So it's attached to this tyrosine kinase, and it activates its own battery of protein this way, as opposed to protein kinase A, the one that actually we never mentioned, but it activates phosphorylates serine and purine. So here's what half of that receptor look like. And so this is where the hormone binds, let's say insulin. But it cannot bind because really the other half has to be there. And the only way that happens is for them to meet on the membrane and then make that part available for insulin to bind, for the weak bonds to happen and for the structural changes there. But once that happens, once they are close to each other, they're too close to each other. And one of the things that happen when those kinds of proteins are close to each other, they start to cross-phosphorylate. Cross-phosphorylate. All right? What is that cross-phosphorylate? This is, by the way, the subtleties of the structural changes. Okay? Personally, if I look at those two structures, I'm not going to be able to say what happens. But really, if you look here, you can see this alpha helix changes its direction from vertical to horizontal. And other changes. And it's because of those changes, again, those weak bonds, those weak changes that happen, we get those messages. But what happened? Cross-phosphorylation means this beta subunit will phosphorylate this beta subunit, and this beta subunit will phosphorylate the other one. They're just too close to each other. They have to cross-phosphorylate. Okay? Once this cross-phosphorylation happens, they are active. Phosphorylation is activation in this case. And so all of a sudden, somebody else takes notice of all these phosphorylation events that are happening, and that's called the insulin receptor substrate, or IRS. That will come and bind to the phosphorylated. It will not bind to the unphosphorylated. But because of that cross-phosphorylation, that happened because they got closer to each other, that happened because of the insulin bound, there is that cross-phosphorylation. And so IRS took notice and bound to the phosphorylated thing. Now what do you think happened to it? It too got phosphorylated. Right? It's a phosphorylation test. And so now it's phosphorylated as well. What took notice of that? Well, this protein here. It's another kinase. Okay? This protein, the phosphoinositide kinase, took notice of the phosphorylated IRS and got activated, itself got excited, and it plucked one of the molecules on the membrane. And that molecule is called PIP2, phosphoinositide phosphate, has biphosphate, and it phosphorylates it to PIP3. You don't have to memorize the structure. So we go from PIP2 to PIP3, from diphosphate to triphosphate. Now that molecule, still in the membrane, and it can travel through the membrane, it's our secondary messenger. Not secondary. It's actually a membrane molecule. Now that can swim gingerly in the membrane as a PIP3. And PIP3, it will be recognized by another protein called PIP3 dependent kinase. Then, PIP3 dependent kinase got excited to find PIP3. Now itself will do what? Will phosphorylate again. And the phosphorylation will happen for yet another protein called AKT. AKT is now activated, and that will phosphorylate itself. So how many times I said the word phosphorylate today? Many times. I really don't know. Okay? But you see what happened here? Now, the reason I'm mentioning this is because it will become, as I said, relevant next week when we talk about the integration test. Because those messages, those phosphorylation events, those proteins, can be short-circuited. And we'll see how actually some of our lifestyle choices, for those of us that have these choices, actually can affect these stuff. And hence can affect the entire metabolic traffic. It can change the entire metabolic profile because of our lifestyle, because we can affect these events at the very top of the pyramid of cellular communication. So, very briefly, cross-phosphorylation triggers a big phosphorylation events. IRS is one of them that gets phosphorylated, and because of that, 1, 2, 3 proteins get phosphorylated after the IRS. One of them is the one that produces the secondary messenger PIP3 that activates another one. And of course, by the way, this eventually will, the AKT will activate another kind of protein kinase, not protein kinase A, that will phosphorylate other proteins. Okay, so in the meanwhile, what's being phosphorylated here? Most of those stuff phosphorylated, right? Kinase. So obviously, to kill this message, there's a lot of things. We're going to have to remove those phosphates. really. Unlike the other one, just conformational changes would do, we really have a lot of phosphates to flush out in order to shut this up. And what are the enzyme, or what's the enzyme that removes phosphate usually? What is its last name? Phosphatase. Phosphatase. We knew that was going to happen. By the way, just since we have working time, part of this message for insulin will eventually go activate the GLUT protein that will allow the glucose to be absorbed by the pancreas itself, not just the liver. The pancreas itself will be part of this amazing. Okay, how to kill this? So here's the killing. Tyrosine phosphatase. Remember, tyrosine is the one that phosphatase. Lipid phosphatase and serine phosphatase. 3 phosphatases have to clear that mess. Each one clear that mess for one of those things. So eventually, everything will go back to bold again. Bold, I mean no phosphate. And that will, of course, inactivate everything. And this message, by the way, because we are using lipid phosphatase, we have a special assassin for that one, the HIP3, which will remove the phosphate from tribe and make it die. And it's no longer the secondary messenger. Hence, it will no longer be recognized by PDK. They're the hip-dependent protein kinase. Okay? Serine, tyrosine. Because this one is phosphorylated as serine, that's why we use serine phosphatase. This one is phosphorylated as tyrosine, that's why we use the tyrosine phosphatase.